Okay, so looking at the actual structure of DNA and what its double helix looks like. So the double helix of DNA has two strands and they run in opposite directions. So you'll see in this picture here, the 5' strand goes down in pink. I highlighted it in pink down to the 3', and then it's the opposite. So now we have 3' going down to 5'. So their opposites sides match each other. Two strands are held together by hydrogen bonding interactions between the bases. So that's what these hydrogen bonding interactions are here. They have complementary base pairs. So A, or adenine, is always going to pair with T, thymine. And the reason is because of the hydrogen bonding. So these are perfectly lined up to have these two hydrogen bonds. And then G and C are going to be complementary base pairs because, again, they are perfectly lined up to have 3 hydrogen bonds now, which is more preferred than 2. So that's the overall structure of the secondary structure of nucleic acids, or DNA in particular. So the main things to remember is that the two strands go in opposite directions, and they're held together by hydrogen bonding interactions. So let's do some questions real quick, or some examples of questions that you might have. So why are GC base pairs more stable than AT base pairs? So GC is more stable than AT. Well, it's because it has more hydrogen bonding. So hydrogen bonding, it's kind of like having, it is having an extra bond, right? Instead of having 2 rubber bands holding something together, there's 3 rubber bands holding something together. So more hydrogen bonds means it's more stable. And then complete the following base sequences in a complementary DNA segment if a portion of the parent strand has the following base sequence. So I gave you in black this base sequence. It's going to be 5' to 3'. So the complementary DNA sequence is going to be where you use the opposite base pair. So I would just write in 3' on this side because we had 5' here. The complementary base pair for C is G, for T is A, and I'll just go all the way down the line, switching those out, making sure that I use T instead of U because we're in DNA. You'll see where that's important coming up soon. So both of these are examples of that. Go ahead and do them on your own. I have these answers written in, but try those out on your own to make sure that you understand which pairs go with what.